Bismillahirrahmanirrahim. Today's topic is about pathology, cellular injury, and these two pages of cellular injury are important. First is related to cellular adaptations, like whenever cells are under stress, how they react. The other is two types of cellular injury, reversible or irreversible. So first we'll go through page 206. Some other terms like aplasia here is added down because it is related to these topics. First of all, we will do them in pairs uh, or in uh, three group of threes. Uh, for example, hypertrophy, hyperplasia, and atrophy. It is the first group because all three are interrelated. Uh, Hypertrophy and hyperplasia, the main difference is the way it takes place. The question asked about hypertrophy is its examples. In hypertrophy, increase structural proteins and increase in the size of organelle ultimately takes place. And we know a very famous example of skeletal muscle hypertrophy in case of bodybuilders. So the question asked is most common hypertrophy of body example is skeletal muscle hypertrophy in case of bodybuilders, and this is a physiological process. The muscle will compensate, and because cardiac as well as skeletal muscles, they cannot divide much, so they are increased in size. That is also important to understand that certain cells, those who do not have regenerative capacity, they increase in size in response to stress. So example asked is about skeletal muscle and also about cardiac muscles. Increase in size of cell, examples of cardiac hypertrophy, that is very famous left ventricular hypertrophy seen in hypertensive patients. If there is hypertension, the heart will try to pump against pressure, and it is just like the bodybuilding of the heart. And but this is pathological because there is hypertension. So question may have stem related to pathological hypertrophy, then it is example would be cardiac myocardial hypertrophy secondary to hypertension. And if it is a most common cause of example of hypertrophy or physiological one, that would be in the skeletal muscle. So two types of muscles, two types of hypertrophy. Moving on to hyperplasia and hypertrophy has a difference, that in hyperplasia cell number will increase, so those cells that can divide, they will keep dividing and making new cells. uh, in very famous example of endometrial hyperplasia, and if endometrial hyperplasia is more, that may lead to benign tumors like leiomyoma. So this is a tumor, but it has hyperplasia as its pathology. So example uh, can be asked that leiomyoma is hyperplasia, or otherwise leiomyoma is not hyperplasia, it is an adenoma. So here the difference between hyperplasia and adenoma or benign tumor is adenoma has capsule. That is also a question asked. So leiomyoma is an adenoma. Strictly speaking, it has a capsule and it is different from hyperplasia because of capsule. But leiomyoma has a hyperplasia inside it if we ignore the capsule. So this can be fit into this example because it is asked in its examples. Other is breast hyperplasia in case of pregnancy, that is a physiological hyperplasia. But the other question, uterine hyperplasia in pregnancy, that has both factors of hypertrophy as well as hyperplasia. So hypertrophy of two types of muscles, skeletal and cardiac, hyperplasia of smooth muscles in leiomyoma case, and also in case of uh, uterus in pregnancy, but that has both hypertrophy as well as hyperplasia. And then moving on to atrophy, that is understood, that is the damage leading to decrease in number of cell or tissue mass overall. For example, there can be multiple reasons. One very common reason is we use POP, plaster of Paris, in case of fracture, that leads to disuse of that arm or leg, ultimately leading to disuse atrophy. So this term of disuse atrophy is important and is asked in example of POP after fracture. And then most common cause of permanent atrophy is asked, that is the denervation injury. So disuse and denervation, two D's in atrophy. Most common cause of permanent atrophy would be denervation, would be better choice, and most common or in otherwise example is POP fracture. And then other causes include loss of hormonal stimulation, for example estrogen and progesterone. We can see breast atrophy, uh, and that can be asked. And then coming to the metaplasia and dysplasia, these are the cellular changes of shapes. In metaplasia, replacement of one type of cell takes place with another type, so it can be asked in this way. Or just like we read about metamorphosis, that is shape change taking place in butterflies. So larva pupa stages, metamorphosis, this is metaplasia. The cell changes their shape. For example, if someone is a smoker, and normally we know that the respiratory epithelium is lined with ciliated columnar epithelium or pseudostratified ciliated columnar epithelium, strictly speaking. Then after chronic irritation, for example by smoking, we know that our outer skin, that is stratified squamous, is resistant to irritation. So the same metaplasia takes place in along the lining. So this columnar epithelium changes to stratified squamous, and this is asked. So the patient or the person having smoking will have both the epitheliums inside, the normal ciliated columnar epithelium or pseudostratified, strictly speaking, and replaced a new epithelium, stratified squamous, that is to counter the irritation or the stress. And the other example opposite to it is Barrett esophagus, that lower portion of the stomach, if chronically irritated by gastroesophageal reflux, gastric acids, and we know that gastric acids are present normally in stomach, and stomach has columnar epithelium, that is the gastric epithelium, and esophagus has the otherwise stratified squamous epithelium, the continuation of skin epithelium inside the esophagus. So the change that should take place to counter this gastric irritation is a conversion of stratified squamous in the esophagus to gastric epithelium. So there are elongated cells which can counter the acid irritation. Outside in the smoker case, it was opposite of it. So this gastric epithelium formation in the esophagus would be called Barrett esophagus, and it is an example of metaplasia, just opposite of smoker epithelium, metaplasia. So most common cause of metaplasia is irritation. Two examples given. Then moving on to dysplasia, that is a precancerous condition. So we usually move from hypertrophy, hyperplasia, then metaplasia, then dysplasia, and ultimately carcinoma in situ and carcinoma. Precancerous condition is mostly dysplasia compared to metaplasia. Then metaplasia is more precancerous than hyperplasia. This precancerous condition of dysplasia mostly takes place in the epithelia, just like metaplasia is usually in the epithelia. And a very famous example of dysplasia is cervical dysplasia. Cervical, in pap smear case, scenario is asked. Human papilloma virus infection causes cervical dysplasia, and coilocyte cells are seen. So, what are these coilocyte cells? They are basically cells that have different shapes, and loss of uniformity of cells and shape, that is pleomorphism, is seen in these cells. And other features, the loss of tissue orientation, nuclear changes like increased nuclear to cytoplasmic ratio, because they are very dividing cells, so nucleus size is increased. But mostly it is the pleomorphism that is asked, or change of cellular shapes and orientation. And this is different from metaplasia, in which one epithelium is totally replaced or partly replaced by the other. And here dysplasia, the cells are the same and they lost the uniformity in them, and some are bigger, other are smaller cells. And they, in case of cervical dysplasia, they are called coilocytes. And now aplasia is different from atrophy in the sense that aplasia is total absence of cells. Sometime during development, certain cells are not produced, the aplastic cells. And in case of example, red blood cells aplasia, rbc aplasia takes place in bone marrow, so bone marrow do not produce that line of cells. So these are the questions asked from this page. In the next page, we are going to discuss cellular injury in sense of reversibility and irreversibility. We know that if cells are not modifying according to stresses like these adaptations, then they will suffer either the reversible injury that is repairable, or irreversible injury that is usually not repairable, and cells die out. First two MCQs here. Glycine amino acid deficiency causes cell injury. So this can be a cell injury related amino acid is glycine. And most common cause of cell injury in our country is malnutrition. We know that. And most common cause of under 5 years death worldwide as well is malnutrition. And now reversible cell injury and irreversible cell injury, all changes will be reversed. First of all, whenever there is injury, for example, blood loss or occlusion of artery, there will be less ATPs produced in that specific area, and less activity of sodium potassium ATPs will take place. More calcium will be inside cells, and there will be cellular swelling. And normally, these pump, sodium potassium pump, working under ATP, they pumps out potassium, and they pump in potassium against the concentration gradient, and pumps out sodium against the concentration gradient both. So more sodium will be moving inside against along the concentration gradient if ATPs are less. So this mechanism is asked, and if more sodium moves in, more calcium will move in, and water along with sodium, it will cause cellular swelling. So mechanism of reversible injury is asked. See, that is the cellular swelling, how it takes place. That is the earliest morphological manifestation. That is another MCQ. But if they specifically asked about earliest ultrastructural change, that is the endoplasmic reticulum swelling, ER swelling. That is even taking place before cellular swelling sometimes. So, and that is ultrastructural. But in case of general question, the most earliest morphological manifestation is overall cellular swelling or reversible injury. And earliest ultrastructural word is used, then it is ER swelling, endoplasmic reticulum swelling. And if specifically about cardiac cell is asked, then it is the contraction bands in cytoplasm. So these are three MCQs. Cellular swelling is overall earliest morphological manifestation. Earliest ultrastructural is ER swelling, and in cardiac cells it is the contraction bands, that is the earliest change observed. And then ribosome will detach from the endoplasmic reticulum and leads to decreased protein synthesis. This is also a reversible change. And then plasma membrane changes like blabbing, not the rupture. Till blabbing it is okay, and just like cellular swelling, it's blabbing, it's a reversible change. Also asked. Then myelin figures, these are aggregates of peroxidized lipids. They are feature of reversible cell injury. They do occur in irreversible as well, and they increase in uh, quantity in irreversible. But specifically, if asked, they start forming in reversible cell injury. So we have to memorize these and learn the mechanism of this sodium potassium pump decrease in ATP. The irreversible cell injury, whenever there is a damage to cell and it leads to rupture of the plasma membrane, then the cytosolic enzymes, like in case of cardiac injury, post MI, troponin will leak out, and calcium from outside will move into the cell that triggers certain enzymes, calcium dependent, and that leads to permanent cell injury. But the question is related to the plasma membrane. Breakdown of the plasma membrane. Here it was just swelling, and three questions related to swelling and contraction bands. Here it is most important sign in is plasma membrane breakdown, and then calcium entry. It is the second most important sign. Most important overall is plasma membrane damage, and then mitochondrial dysfunction, loss of electron transport chain, uh, and then a rupture of lysosomes inside the cells. We know uh, lysosomes have enzymes for digestion, so instead of digesting out other things, they start digesting inside the cell. That is called autolysis. So apoptosis, or programmed cell death, and autolysis both are feature of irreversible cell injury. Membrane damage, autolysis, apoptosis, as well as necrosis, these are all asked in MCQ. These are all signs of irreversible cell damage, because now cell cannot go back. It is digesting itself, or it is necrosed, or it has triggered apoptosis, that is programmed cell death. So death is irreversible thing. And most reliable sign of autophagy is the rupture of lysosomes, because whenever they are going to rupture, they will release enzyme that will digest the cell from inside, that is autophagy. So most important sign of irreversible cell injury is plasma membrane breakdown. Most reliable sign of autophagy fast, it is rupture of lysosomes. And then a nuclear degradation takes place inside the cell, that is the brain of cell dying. So that is the main feature of irreversible death. Nuclear degradation, first it is the nuclear condensation, that is pycnosis, and then it is karyorrhexis. Nuclear, first it will be pycnotic condensed nucleus at the center, then it will be karyorrhexis, that is nuclear fragmentations, and in case of apoptosis, these are specific ladder fragmentation that is asked. We'll cover in apoptosis. Karyorrhexis, and then ultimately leads to karyolysis, nuclear dissolution. Nucleus will be totally invisible. So the DNA damage is in this sequence: pycnosis, nuclear condensation, then fragmentation, and then dissolution. So all these three terms are asked. Pycnosis is a feature of irreversible damage. Karyorrhexis is also karyolysis ultimately. And then amorphous densities, inclusions in the mitochondria are seen. These are flocculent densities or amorphous densities inside the mitochondria seen in irreversible damage. Now mitochondria has stopped totally working, so ATP won't be produced at all, and that is called loss of electron transport chain. In irreversible, there were decreased ATP just because less oxygen is going and there is cellular swelling. If this reaches a limit, that these autolysis, autophagies, and karyolysis are triggered, then it is or plasma membrane damage is there, then it is irreversible. And you can learn about them in this layout of cells. Normal cells, reversible is blebbing, that is most swelling or blebbing. Then irreversible is rupture of lysosome and autolysis, and ultimately cell death. Hope these two pages will clear doubts about these topics. Thank you.